PhD student Oliver Vining is collecting samples for biomedical research at Axial Seamount. Here you see his syringe sampler in the closed position and then in the open position. Oliver Vining explains.
This is Jason taking a suction sample of some sediment at the anemone vent, and you can see that the syringe was just fired by the hydraulic thumb on Jason's right arm pressing down on the firing pin of the syringe, which released the springs and pulled back the plunger in the chamber. So now you can see that the chamber is filled with white microbial mat and sediment. So the reason that we're sampling this spot in particular is that the bacteria that we're trying to isolate or collect are actinomycetes, which don't necessarily like to live in hot water. They like to live in cool to warm water, about maybe 10 degrees. So you can see the shimmering hot water coming out of the middle of that vent. That's probably between 20 and 30 degrees. So we want to sample just outside of that shimmering water. And now you can see the ROV pilot putting the syringe back into its holster. He wants to put it with the tube sort of pressed against the side of the chamber so that it seals off, so that we don't lose the sample falling out of the tube at the end, the nozzle, or have it be contaminated by the surrounding seawater once we bring Jason up to the surface.
The syringe sampler is further secured with a bungee cord manipulated with Jason's agile arm. This attaches the sampler to Jason's payload, where it will remain for the rest of the dive.